Well, good afternoon. As you heard, I'm Steve Rosenberg. I'm the Chief of Surgery at the National Cancer Institute, a position I've had for the last 42 years. I've been working on immunotherapy since that time. Let me just put the framework of today's discussion about immunotherapy in the context of cancer treatment as a whole. As you know, we've had 3 effective treatments for cancer: surgery, radiation therapy, and chemotherapy. Surgery goes back 3,000 years to the Egyptian papyri. The radiation therapy dates to just a year after the discovery of X-rays by Röntgen in 1895, and modern chemotherapy dates to chemical warfare research here in Maryland that showed that nitrogen mustard could, in fact, cause the decrease of lymphomas in patients. And all modern chemotherapy dates from that accident in a laboratory in which laboratory technicians were exposed to nitrogen mustard back in 1942. Well, the best application of surgery, radiation therapy, and chemotherapy will cure about half of all people who develop the disease. And so in many ways, cancer is quite a curable disease, but the half that cannot be cured, last year in the United States alone accounted for a little less than 600,000 deaths. 1 in every 3 of us will develop an invasive cancer during our lives, and unless we find better ways to treat it, 1 in every 6 of us will die of cancer. And so we desperately need treatments to join surgery, radiation, and chemotherapy. And we now have a 4th arm to this 3-legged stool, and that is immunotherapy. It's different in concept from radiation or surgery or chemotherapy that apply external forces to treat the cancer: a scalpel, a radiation beam, or a drug. In immunotherapy, we try to stimulate the body's own natural immune system to defeat the disease, because the immune system recognizes the cancer as foreign, but not foreign enough to eliminate it. And if we could stimulate this immune reaction, as you'll see, we can cause the regression of even major amounts of cancer. Now, how did I get started in this? It was actually when I was a junior resident in surgery at the Pete Brigham Hospital in Boston. I was rotating through the West Roxbury VA Hospital. This was in 1969. And I was covering the emergency ward, and a patient came in. It was a 68-year-old veteran who had had right upper quadrant pain. And it looked like he had a typical gallbladder attack, and it was one of the first actual operations I did as a junior resident to remove his gallbladder. But when I went through his chart in the emergency ward, I saw this absolutely amazing story. The patient had been seen in that emergency ward 12 years earlier with a lot of abdominal symptoms. He'd been worked up. It turned out he had a gastric cancer, a cancer that started in the stomach. He had had an operation. I turned the pages on this chart and saw that he had gone to the operating room, and there was a surgeon's note saying tumor had spread through the belly. There were liver metastases. I biopsied one. It was the cancer had spread to the liver. There was nothing we could do, and I closed the abdomen. We gave him an appointment to see us in 3 months, but nobody expected to see that patient again. Well, sure enough, 3 months later, the patient came to the emergency ward, and 6 months later, he was gaining weight and doing well. A year later, he came back, and he was back at work. And here he was 12 years later, perfectly normal, with the exception of gallstones. I operated on his gallbladder, took it out. And in fact, his abdomen was completely free of all cancer. Went back and checked the old biopsies. Sure enough, it was cancer. It was the same patient. This patient had experienced one of the rarest events in all of medicine, and that is the spontaneous regression of a cancer in the absence of any treatment. In fact, it was the 4th reported case of this in one of the first papers that I wrote in the medical literature. But somehow, this patient's body had learned to destroy his own cancer in the absence of any treatment. And I've spent the last 40-plus years or so trying to figure out how that happened and how to make it happen more regularly. Well, I came to the NCI. I finished my residency on June 30th of 1974, became chief of surgery July 1, 1974, a position I've held ever since, and began working on this problem. How could we stimulate the body's immune system to fight the disease? Well, a molecule called interleukin-2 had been described, which was called T-cell growth factor. It was a way to stimulate T cells in the body. And we had been learning from experiments in animals that it was, in fact, the T-cell arm of the immune response, not antibodies, but T cells that circulate in our body. We each have about 1 million of these circulating in every milliliter of blood, every thimbleful of blood. And it was these T cells that appeared to be active in animal models to cause cancers to regress. And so I began studying interleukin-2, giving it to patients in different schedules. We treated 66 patients in a row, all of whom had advanced cancer with varying schedules and doses of interleukin-2. None responded, all would go on to die of their cancer, until finally the 67th patient, a woman I know very well who was 33 years old at the time, had widespread melanoma. We finally used a very high dose of interleukin-2 in a given schedule, and her cancer disappeared, and 30 years later, she's completely normal. When we see the complete disappearance of cancer in a patient with immunotherapy, it very, very rarely ever comes back. Well, interleukin-2 is one way to stimulate the body's immune system, by directly stimulating immune reactions with the hope that a natural immune reaction against the cancer would then lead to the cancer disappearance. And in fact, in patients who receive interleukin-2, in about 15% of these patients, we will see dramatic regressions, and about 1/3 of those will see a complete disappearance. But it's a low number. It's only about 6 or 7% that would be cured with interleukin-2. But there are many other ways to stimulate the immune system, and very recently you've heard a lot about checkpoint modulators. Virtually every physiologic system in the body has something that stimulates it, something that inhibits it. We have a sympathetic nervous system, a parasympathetic nervous system. We have hormones that will increase acid secretion in the stomach, hormones that will decrease it. Well, the immune system is the same. Interleukin-2 is an enhancer, but checkpoint modulators such as ipilimumab or anti-PD-1 enhance immune reactions by taking the brake off immune reactions. That is, there are immune reactions that inhibit immunity, and anti-PD-1 and ipilimumab can prevent those from inhibiting and thereby enhance immune reactions. Well, there's a second way to do immunotherapy, and that is cancer vaccines, a very active area of investigation, but as yet we've not figured out a way to directly immunize patients against cancer, but it's an area of active investigation. The third area of immunotherapy is the one that we're talking about this afternoon, and that is based on the fact that we know that T cells, especially T cells stimulated by interleukin-2, can mediate the regression of cancer in some patients. Well, why not try to use T cells themselves as a drug to treat cancer? If in fact there is a T cell response in the body that recognizes cancer, if we could identify it and isolate those T cells, then we might be able to give them back to the same patient and cause cancer regressions. And it's that whole area of cell-based therapy that's now in an explosive realm of progress. So I want to show you just a few slides to finish off my few comments. I just emphasize in this slide that there are two parts of the immune system, antibodies and T cells, but it's the T cell arm of the immune response that is capable of mediating immune reactions. I show this scanning electron micrograph, which is not a cartoon, it's a real photograph, magnified 1 million times, of the battle that's going on in our bodies when we stimulate immune reactions. You see here, covered in white, are lymphocytes. This is actually a melanoma cell in culture. We've raised lymphocytes that can recognize this melanoma cell based on unique antigens that it can express that are not present on normal tissues. And when we give T cells to patients, that can recognize a cancer, they seek out the cancer, attach to it through these small pseudopods that have a T-cell receptor on them, and inject materials into the cancer cell that can destroy it, but they also make other molecules that call other cells into the battle, macrophages and neutrophils, other kinds of white cells. Well, we've studied over the years several ways to make good T cells to treat patients with melanoma and other cancers. And now in patients with melanoma, in almost 60% of patients, we can mediate regressions of cancer by giving patients back their own T cells. Now, there are two ways that we do this. One is to take a patient's own natural cells from a tumor. We call them tumor-infiltrating lymphocytes. And intuitively, what better place to look for a lymphocyte doing battle against a cancer than within the cancer itself? We take that tumor out, and as you'll hear from Jamie, this is exactly the kind of treatment that she received. We isolate those cells that were inside the tumor. We have ways now of using in vitro techniques to identify the exact cells that were reacting against the cancer. Sometimes there'll be 0.001% of all of the T cells, but we have ways to recognize them. We grow them up to large numbers, and we infuse them with interleukin-2. And it's very important that we first wipe out the patient's own immune system before we do this, so that we can replace it with cells that have anti-cancer activity. And when you do this, and I'll just show you a few quick examples. I'm not going to show you Jamie. She'll talk about her own case. But what we do is, again, take out the tumor, isolate the immune cells, grow them up, and give them back. And so here's an example of a patient that I'm showing you to make the point that there's no relationship in immunotherapy and T-cell therapy between the amount of cancer that you have and the likelihood of having a complete elimination when you find the right T-cell that can react against an antigen on the cancer. And you can see this patient who had cancer throughout the liver, the liver. Every one of those, this is a CAT scan, so it's a transverse cut through the body. And what you're seeing here is the liver and the spleen. Every one of those dots is a cancer deposit that were increasing with time. On day 0, we took out a cancer, isolated the T cells, treated the patient on day 0. By day 34, almost everything was gone. A few things remained. But this patient remains disease-free now 12 years later of all of these liver and lung metastatic deposits. So there's no relationship to the amount of tumor and the likelihood of getting a complete regression. It can happen very fast. This patient's subcutaneous disease disappeared in just 12 days. Sometimes it takes longer, but you'll hear a little bit from Jamie about her unique experience with this kind of tumor. And most importantly, the brain, which can be a problem, is not a sanctuary site for T-cell based therapies. If you can get a T-cell from a patient, give it back to that patient as a drug, then tumors in the brain, as you can see here, can be made to disappear as well. Well, the second kind of T-cell therapy, and I'll finish with this, involves the genetic engineering of T cells. And we have one of the world's experts here, Dr. Brentjens. You're going to be hearing a little bit more about this a little later. In this kind of treatment, and again, Jamie was part of the history of the development of this, which is why I mention it, we take out a patient's own cells. We can get them from peripheral blood or the tumor. And now we can genetically modify them by introducing new genes into the lymphocytes to give those lymphocytes properties that have never existed before in the course of evolution. You can take virtually any gene from any species, put it into a human cell with high efficiency, get it expressed in 90% of cells, and give those cells now new properties. So we have the ability to manipulate the immune system in quite remarkable ways. And one of the ways we do that is by putting in receptors here against this is a lymphoma treatment you'll be hearing about from Dr. Brentjens, where we take a particular kind of receptor that a lymphocyte doesn't have. We put it into the lymphocyte, and we can, with this kind of approach, cause major amounts of disease. Again, this is a CAT scan through the chest, and you can see that disappearing, these large lymph nodes disappearing. and these disappearing as well. Every one of these, in this PET scan, every one of these dots is a deposit of cancer that can be made to disappear by getting these genetically modified cells, as did these liver tumors disappear. Well, this final slide is sort of a prelude to talk about Jamie's case. Because TIL cells, tumor-infiltrating lymphocytes that we isolate and give back to patients, can cause regressions in about 60% of patients, and now 25 to 30% will have complete disappearance of all their disease permanently, that is in a durable sense. But we were learning as we did this back in 2010 and 2011 when we first met Jamie, that in fact we could improve the ability of TIL to work by introducing into those TIL a gene that causes an inflammatory response at the tumor. And that gene is a gene called interleukin 12. And we had shown and published that when TIL didn't work, when we gave 3 times 10 to the 10, that is 30 billion cells, when we put in that gene for IL-12, which causes inflammation in the tumor as well as attacking the tumor, we could, with 1,000-fold fewer cells, cause complete regression, as we did in this patient who remains disease-free now, over 4 years later. And so when Jamie came to us, we used what we thought was the best we had available at that time, and that is T cells that we could obtain from Jamie, genetically modify them, and give them back. That's a whole new form of cancer therapy, using cell transfer, not only with natural cells, but with genetically engineered cells. And the first time that was done was in 2006 in our group in a patient with melanoma, using a different kind of modification. But it's a very new approach that I think has enormous application, not only to the treatment of melanoma, but other cancers as well. Well, with that, I'll turn to Jamie and ask her to tell you all about her experience with this, what was then radically new treatment, but today is something that we do almost daily in terms of giving patients genetically modified cells. So, Jamie, why don't you tell the group a little bit about what happened to you?
Yes, Jamie Goldfarb. I'm the privileged recipient of all of this brilliance here, thankfully. But I was diagnosed stage 1 in 2007. It was a birthmark on my leg that changed over time. And I know some of you in the room heard my very detailed story last year, so I won't go into quite as much detail now. But it was a stage 1 that was in my leg, and my dermatologist said, you know, I have a friend who's a medical oncologist. You're young, you're going to probably start a family soon or go about your business. You should be followed by him just to have someone following you, because who knows what can happen. So over the course of the next year, after I had the primary removed, I had a wide excision, everything was clear, I had clear scans, but I kept getting recurring infections in the surgical area, and my surgeon kept sending me away and saying, this has nothing to do with me, get it treated by your primary care physician, and stop calling me, basically. And after the 4th infection, my medical oncologist called him and said, you need to see your patients. So they went back in thinking maybe that they had left a suture behind that was causing infections, and what they found was a massive melanoma in the deep tissue. So then I was stage 3, but I had just gotten married, and there wasn't, at the time that I was diagnosed then, it was 5 years ago, that was 2010, actually, and they said, we don't really have any recommendations, we'll watch, just go about your business and go on with your life. So we did, and the next month I got pregnant with our son Kai. So then I was pregnant, didn't have anything, didn't do anything, didn't have any scans or anything. And the week before I was supposed to go back to work for maternity leave, my medical oncologist said, let's do a scan just because we haven't done anything and you've had a baby and now you're going back to work. So we did that, not expecting anything to come up, and that scan showed melanoma in my liver and my pancreas. So then at that stage, I was stage 4. When my medical oncologist came in to tell us the results of the biopsy to confirm it, he said, you know, we're going to do everything we can to buy you time, and the best way I can buy you time is to send you to somebody else, which was a very forward-thinking medical oncologist, and I'm thankful for that decision every day. But he set up an appointment immediately at NCI so that I could screen for one of Dr. Rosenberg's studies. And at the same time, we also consulted with experts at University of Pennsylvania and Mass General to get all the opinions that we could for the different research that was going on. Because when I was diagnosed, there were only 2 approved treatments for melanoma. One was interleukin-2 and one was chemotherapy. So one, really. So everything was in research, though. Ipilimumab was in research, the BRAF inhibitors and those kinds of things, and the TIL trials. So we went to NCI and screened, and at NCI, the way that the treatment goes is there's Dr. Rosenberg, then there's a team of attending physicians who are full-time staff, and they rotate and when they're seeing patients. And then under them is a group of research fellows that rotates out every year. But as a patient, your most contact is with your research fellow. So when we went to screen, we met my research fellow at the time, who was Dr. Schaub, and we took with us a picture of me and the baby, because we knew that he was going to go back then, and you were going to discuss as a group what the treatment decision should be. And we wanted him to be very clear that our eye here was on the end game, that it didn't matter what was going to happen in the interim, but we had this baby and I needed to be alive for him. And so Dr. Schaub went, took the picture, you had your meeting, and then called and said, we think you should do the TIL trial. So I went back to NCI, had a core biopsy of my liver to extract some of the tumors that were in my liver so they could grow the new cells, as Dr. Rosenberg described. But in the meantime, that process takes about 6 weeks. So in the meantime, they decided to administer high-dose interleukin-2 at NCI. So I did 4 rounds of that, 27 doses total. Happy to answer any questions on what that's like as a patient, but it can certainly be very worth it. And for a while, it shrank the tumors in my liver and my pancreas. And at the time, I was being scanned every 4 weeks. So my last interleukin-2 dose was April of 2011, and then the scan showed the tumors were shrinking until the September scans, when all of a sudden 30 new subcutaneous tumors popped up in a 4-week period. So then we decided to proceed with TIL. And I did the trial that Dr. Rosenberg was describing with the genetic modification of the interleukin-12. So it was about a month-long inpatient. And when I was checking in, we were putting up pictures of the baby. And the nurses kept coming by, and they were saying hello to us, and they knew us. They knew our names. They knew the baby. We didn't know them at the time. And the first couple times, we just thought, wow, this is great customer service at the hospital. And then the 5th time it happened, we said, why does everybody know us? Why is this normal? Do you just know everybody like this? And she said that the reason was because someone from the team had taken the picture that we had given Dr. Schaub and walked it all around the unit and said to the nurses and everyone else on the unit that we would be checking into the hospital and that they were going to save my life. And so we started our whole journey with 100% confidence in the medical team and every decision that was being made along the way, and not just because of the 42 brilliant years of immunotherapy pioneering, but because of the way that Dr. Rosenberg and the entire team treated me as a whole person, not a research subject, not just a cancer patient, a whole person with an understanding that I had just created a life and had a responsibility to stick around for him. And my last treatment, so I did the TIL in September of 2011. It was about a month-long process. So I checked in to the hospital, then had 5 days of intensive chemotherapy to kill my immune system, then waited for the counts to go all the way down to 0, complete neutropenia, and then the new cells were introduced. And then we just waited for the counts to come back up. Because of the genetic modification, I did not have IL-2 on the back end of that treatment as you would in standard TIL, so we just waited for the counts to come back up, and then went home. But that was about a month. And then that was September of 2011. Scans in September, October, November, and December showed that the tumors were growing. So it didn't seem that it was working. And in the interim, took out a large tumor that was attached to my right ovary in an attempt to try to grow more TIL. It didn't grow. Thought maybe it was too close to the chemotherapy and the cells were lazy. But my attending physician at the time said, okay, well, next visit, we need to start thinking about what we're going to do next. What's your next steps? And I kept saying, I'm not ready. I'm not ready to make another decision. That was a really complex treatment. Let's just give it a little bit more time. In December, I got an upper respiratory infection. And during that time, I could feel physically my tumor starting to shrink. And then my January scan showed that the tumors were shrinking. And then they shrank steadily over the next 2 years until about 2 years after that, there was no evidence of disease. And there's been no evidence of disease for the last 2 years. So.
So the cells that we give are different than conventional treatments. What we use with cells is a living treatment. The cells that we give are yours, they're alive, and when you get immune cells, they stay with you as long as you're alive. When you get immunized against smallpox, for example, as an infant, then you still have those T cells in your body 60, 70 years later. And so what we've learned is when we give these cells, they can expand as a living treatment 1,000-fold in the first week after they're given. It's not like surgery, you close the belly, you're done, hit the off button on a radiation therapy machine, it's over. These are cells that continue to work. And they can sometimes take time to work. And so what was happening inside your body is we got samples and could study them as you came to clinic. We would always draw some blood and save serum, and we cryopreserve lymphocytes so we can go back and do studies. And we learned so much from Jamie that helped not only Jamie but other people as well. Because what we found was that those cells stayed alive, they were circulating through the body, and over the course of months to years can cause tumors that arise to react against these lymphocytes and shrink. And when we see complete disappearance, as we have in Jamie, as I was just telling her a few moments ago, we've almost never seen the cancer come back. When you can induce an immune reaction that causes a complete disappearance of the cancer, it's generally curative, which is a lot different than that of most chemotherapies for solid cancers, which can cause transient regressions, but then the tumor will regrow. Now, I know the treatment was difficult for you, but you were courageous in taking it. What was your attitude? And I'm sure we explained to you we were doing something new that hadn't been done very many times with this interleukin-12.
Yeah, the interleukin, the TIL process was, going down to zero to neutropenia and having the front part of the 5 days of the high-dose chemotherapy. It's difficult, but it was not nearly as difficult as the interleukin-2. So I'm glad I did that treatment first, because after that you could do anything after IL-2. But
right, the interleukin-12 and interleukin-2 interact in ways that you just can't use. You can't use both. But tell me about your clinic visits. Patients will often tell me that coming back to clinic to have their scans when they don't know what's happening inside their body are some of their most difficult moments. Tell me a little bit about that.
Yeah, even the waiting between having the scans and finding out what the scan results are is a very difficult time of life. In general. I mean, it's getting easier certainly as time goes by, but the fact that somebody knows somewhere, even if it's just a computer at that point, knows if you're going to continue to live or if you're on your way to dying is a very difficult time. But going back to the NCI is like a homecoming. I look forward to my clinic visits. The doctors, your team, and it was part of my family now. I mean, I have spent my research fellows when I was inpatient we were the same age at the time, and so they would come in and they would hang out with us. And Dr. Schaub would come in on the nights that he was staying overnight, and he would watch TV with me until midnight and scratch my back and those kind of things. And some of them, I'm still friends with a lot of them, and some of the nurses are some of my very good friends now. And just the knowing that you were actively trying every single day to save my life is something that I can't, that will stay with me forever. The cancer is gone. The experience of having cancer will stay with me forever in a beautiful way.
That's very kind. And you had actually multiple things you could see disappearing because you had so many lesions in the skin. So that must have been quite a remarkable experience to watch them going away. Most people can't do that. At the NIH, everybody that we treat at the National Cancer Institute has advanced cancer and has been through multiple other treatments and come to us only when there's nothing else available. And when we first saw Jamie, the only approved treatment for melanoma, the only approved immunotherapy was interleukin-2, which got approved in 1985 just for patients with melanoma and kidney cancer. And chemotherapy never had much impact. So when your doctor saw that your melanoma had spread, the NIH was a reasonable place to come, since we were experimenting with these new therapies. A lot of our patients will not call the NIH to the National Institutes of Health. They'll call it the National Institutes of Hope. Many of the patients that we see will tell you that their worst moments are not when they find out they have cancer, not when they've received a treatment that hasn't worked, but when somebody says, "I'm sorry, there's nothing left to do." And one of the things we can do at the NIH is provide, on the basis of the mandate of that institution, which is not to practice today's medicine, but to create the medicine of tomorrow. That's what the NIH does. That's what the NIH funding is dedicated to do. And that can provide hope to patients even today who have cancers that don't have effective treatments available to them. Now, our early work was in melanoma, but that is now being extended quite dramatically, because in the last few years we've learned ways to identify cells that can react against common cancers. And about 2 years ago, we published the first case of a patient who had liver tumors from what we call a cholangiocarcinoma, a bile duct cancer, that had spread throughout her body. We could identify immune cells in that patient, grow them using new techniques to large amounts, and give them back and see a regression. And we're now seeing that in some patients with colon cancer as well. And so this has been a slow evolution of the development of immunotherapy, but we're getting better every day. And now, for the first time, we have blueprints for how to apply immunotherapy to the treatment of other kinds of cancers. Melanoma taught us a lot, as you did, Jamie, in our treatment of you and in the study of all of the cells that we obtained. But that's now being applied for common epithelial cancers like those of the colon, the pancreas, the liver, the prostate, the ovary, and the solid kinds of cancers that still today kill about 90% of everyone who dies of cancer. So we're getting better. Yes, Wendy.
Can you talk a little bit about, you were really lucky, Jamie, to get to Dr. Rosenberg and to go to the NIH, and the NIH is an amazing place, but not everybody with cancer can get to the NIH. Can you talk a little bit about what it's going to take to move these therapies out where they're widely accessible? So look ahead in the crystal ball of how we're going to make these kinds of procedures and therapies available. What do you see coming?
Well, big pharmaceutical companies tend to emphasize the development of drugs in a vial that can be shipped around the world. And it can cost perhaps $500 million to develop the first vial of an effective cancer therapy. But the next vial has to cost $1 or $2 for that to be a commercially viable treatment. And so the kind of treatments that we're talking about, immunotherapy using cellular therapy, are very different. Every time we treat a patient, we have to create an entirely new drug, because it's that patient's own cells which are being identified as anti-cancer cells or are being genetically engineered that then have to be grown up and given back to that patient. And we haven't made a great deal of progress with drugs in a vial, frankly. It hasn't impacted hardly at all on the death rate from cancer, although I think the new checkpoint modulators, at least for melanoma and some rare patients with certain kinds of high mutation colon cancer, are going to be dramatically helped by the checkpoint modulators. We desperately need additional treatments, and I think the use of the body's own T cells is going to be a very important part of the future development of this immunotherapy. Now, the problem is when you have to create a new drug for every patient, it can be not only complex, but expensive. And for many, many years, people felt that cell therapy was not going to be effective. We used our first cell therapies back in 1988 in patients with melanoma. That one couldn't ever develop a cancer treatment that was so personalized. Because think of what we do. We take a patient's own lymphocytes, generate a drug that will treat their cancer and no one else's. And in fact, now we know that it recognizes unique mutations in that patient's cancer that are unique to that patient and not present in other people, which was the case for you too, Jamie. And that is that your cancer had certain kinds of changes within it that were quite unique to your cancer that the TIL cells could recognize. Well, when things work, the genius of American industry finds out a way to get it to people. I don't think anybody who has advanced cancer cares about how complicated the treatment is or how much it's going to cost to develop the drug. They want the treatment. And there are now, for example, with the kinds of genetically engineered treatments for CD19, and you'll hear from Dr. Brenshans, I'm sure, a little bit more, there are now 20 companies worldwide that are trying to develop cell-based therapies for patients with cancer. And these involve not only naturally occurring cells, but cells that are genetically engineered as well. And I'm delighted to see Mark Theoret here, one of the fellows who was in my lab. How long ago, Mark?
Probably 2009.
2009. Mark was working when he was just finishing his fellowship in oncology and hematology, working on some of these genetically engineered cells as well. At any rate, there are companies that are now developing the use of these genetically engineered cells to treat patients with lymphomas. And there are many of them. Some names you've heard, I'm sure: Novartis, Kite, Juno, Celgene. There are companies now that are developing naturally occurring tumor-infiltrating lymphocytes, Lion Biopharma. And one of the roles of the NIH and academic scientists in general are to develop innovations which can then be taken over by commercial companies that are capable of delivering that treatment to people in need worldwide. And I think that's happening with these cell therapies. Yes.
Hi, I'm the creator of a community of cancer patients, and they happen to be colorectal. I don't know how to turn this on.
I can hear you.
Yeah, I'm loud. You mentioned I know a number of the people who've been in the class and who are doing well, thank you so much. The question is, I don't know if this is working right.
Sorry, go ahead.
The question is, you mentioned something about the cells being unleashed, the immune cells and the TIL cells unleashed to go to work. I don't remember the terminology.
Yeah.
Well, one of our survivors' patients was on one of the trials, and she did well until the therapy started to fail her.
Right.
And she was put back on regular chemo. She had a horrible time. She had to be hospitalized. And the speculation that she had, and I wonder about, is are those cells still working and fighting with the chemo that she had? Because she'd had this treatment once before. It was a FOLFOX or a FOLFIRI, something like that. She was really ill. It almost killed her.
You mean the chemotherapy?
Chemotherapy. And I just wonder if the unleashed cell activity hadn't created that. I don't know biology. I don't know nothing about any of this stuff, except that I work in the field today, and I'm also a survivor myself. So I'm always curious to find out, A, why I survived, and why other people do survive as well. But that's my question. What happens after 3 or 4 months?
The patient you're talking about had received the cell therapy, and yeah. So let's
7 or 8 months.
right. So again, we need to understand that these cell therapies, including the treatment that Jamie received, are still experimental treatments. Now, and depending on the kind of disease you have, they can either work commonly or work only rarely. Now we can do very well in patients with melanoma. Almost 60% of patients will respond, and at least a quarter will be cured of all of their disease. But for other kinds of cancers, the cells aren't nearly as potent. In fact, the first time that a cell therapy was ever shown to work in a patient with a solid tumor other than melanoma was this patient I mentioned that we published 2 years ago with a bile duct cancer. So we're just learning now how to treat colon cancers and these other tumors. It's very early in the development, and they're not available in most places. There are only 3 institutions I'm aware of that were capable now of doing the kinds of things that we're doing to develop these treatments for patients with the common solid cancers. And we learn a lot. And the question is, why do some patients respond and not others? Well, I think we finally learned the final common pathway of virtually all immunotherapies, and that is interleukin-2, the checkpoint modulators, ipilimumab, anti-PD-1, TIL cells that Jamie received, and that is that these cells recognize unique changes in the products of DNA inside the cell. When that sperm meets the ovary, the ovum that creates humans, there are 23,000 genes or so that come together to make us what we are, and any one of them can mutate as those cells grow and continue to grow and change as we're adults. And as they grow and replicate their DNA, completely random changes can occur because of mispairing of how DNA can replicate itself. And those random changes are called mutations. And sometimes those mutations can cause a cancer, but they can also be the target of the cancer because they give rise to foreign molecules that the body has never seen before. And one of the things that the immune system can do is distinguish what is naturally normal self from non-self. And that is the mutation, which has been created in the cancer. And some cancers will create mutations that the immune system can recognize very readily, and you can have 0.1% of all your lymphocytes that recognize it, but sometimes 0.010%. And as we get better to identify the exact cells and can transfer them, I think we'll improve the treatment. Now, why does it not work in some people? Because the numbers of immune cells that can recognize those mutations vary dramatically across patients, and some patients have very few and are unlikely to respond unless we carefully select the right cell. But patients that have many of them, it's much easier to find them, and fortunately for Jamie, her tumor had many of these kinds of mutations.
Dr. Rosenberg and Jamie, thank you so much. And as we kind of move into the next panel, you've set the stage beautifully for this. And I wonder, Dr. Rosenberg, you've been in this for decades. Jamie, you've been fighting for 5 years now and successfully. I wonder with the headlines that you're now seeing of immuno-oncology on the front page of every paper and significant gifts being made by likes of Michael Bloomberg, Sean Parker, et cetera, what do you think, just very briefly and maybe leading into the next 2 panels, is missing from those exciting headlines? It's very exciting news for patients and all of us in this room, but as you read those headlines, I'm sure it's exciting to see the work, and I'm sure it's exciting for you, but what one thing do you think is missing that we should all keep in mind as these conversations continue?
Well, we need to remember, again, we're seeing Jamie as a hero, really, as someone who withstood multiple experimental treatments, and then finally the right ones occurred for her. But many patients still don't respond. And although I think of Jamie and patients like her often, I can tell you when I lie awake at night, it's not thinking of patients who responded, it's that large majority that do not respond. And why does that happen? And in a sense, it was the answer to the prior question. The cancer process is immensely complex. And now that we understand, and this is a product of the last 2 to 3 years, now that we understand that the final common pathway of the immune system recognizing a cancer is the recognition of these mutations, that has come about because of the joining of genomic technology, which has been quite stunning. It took 10 years to sequence the first human genome and $2 billion. We now do it in my lab in 2 days, and can be done in labs around the world in 2 days. So we have new tools available that can tell us what the mutations are. I mention this as an example because there's an enormous need for support of basic research, because we never know what realms of knowledge are going to come together in a way that will be profitable for the treatment of patients. And in this particular case, it was the marriage of genomics with high throughput immunologic screening, both of which were developed through a lot of basic science around the world, that has enabled the development of some of these new treatments and emphasizes the need for a lot of basic science as well as translational research. And so what's missing is knowledge. It's ironic that the very mutations that often result in a cancer can be its Achilles heel, can be the targets of effective therapies. But what we desperately need is more funding for not only basic research, but translational research, which is one of the reasons that I agreed to come down here today to thank you for all the work that you do in trying to emphasize the need for additional work to save and to help the 600,000 Americans today who suffer from cancer and will ultimately die of it. So thank you for all of your efforts.
So before we move on to the next thing, I wanted to give Jamie the last word. It looked like you were heading there, and I maybe wanted to tee it up a little bit and ask you to sort of bring us back to the voice of the patient, and you get to be here speaking for all patients today. You have a lot of people in the room here who are leaders in innovation in a variety of different ways. So I'd love to know, I'd love for you to charge the group back from the perspective of the patient. So what would you like to see happen going forward?
So to also answer your question and your question at the same time, what's missing from a patient advocacy perspective from those headlines is an educational component that speaks directly to the patient in a way that a patient can understand. Patients don't yet understand what immunotherapy is and how it's different from chemotherapy. There's a lot of confusion in that realm. They don't know how to access it. They don't know how to go about finding a clinical trial. They don't know the resources that are available, and when they find the resources that are available, like clinicaltrials.gov, they can't understand it at all. It's not written for the general public. It's written for the research community. And what some people in this room, there is a widespread effort in pharma and biotechs to have a more patient-centered focus in clinical trial development and to more fully understand patient needs and desires when designing protocols and informed consents and that kind of information. But we need to take that a step back, and we need to get patients information about clinical trials in general so they can understand what clinical trials are, they can understand how to access them themselves and not be relying on their doctors as gatekeepers, because the conversation between doctors and patients about clinical trials for non-research physicians is not happening nearly as often as it should be. And one of the reasons that people are coming to the NIH so late in the game is because no one is telling them about it. And they're getting to the point where they're finding out from their medical oncologists or other doctors that there's nothing else left for them to do, and no one has ever had a conversation with them about clinical trials, about the NCI, about the existence of other opportunities. So there's a strong need in the patient community for patient-directed information that patients can understand and that patients can access easily.